All right, in this video, we wanted to talk over some details about another one of these biological macromolecules. This one we're going to discuss in this video are lipids. So let's jump right in. So lipids are part of cells. Lipids include things like fats and oils. So this picture demonstrates that. Butter is full of fat, right? Margarine, mayonnaise, and salad dressings are full of fats and oils. So we eat these things because when we eat living organisms, previously living organisms, they're composed of some fats and oils. So here, again, we're showing you some examples. Lipids includes fats and oils, and we're going to see that they're an important part of every single cell. They're going to be critical components of all cells, and we'll be thinking about why as we move forward in the course. All right, so we'll start by thinking about what functions lipids perform in cells. And then we'll look at the structure of the lipid and give some specific examples of lipids that perform each function. So to start, lipid functions. There's three main functions that lipids perform. The first is energy storage. This one's pretty obvious, right? We can think about fat on our bodies. Fat on our bodies is energy that our bodies have stored for use later on if we're short on food. So every organism can use lipids as a way of storing some energy for later. Another function that lipids perform is that they are a critical component of the cell membrane. So as we talk about cell biology later on in the course, we're going to see that lipids make up the cell membrane, and the cell membrane is absolutely required for proper cell function. Okay. The third important role that lipids play is that they are signaling molecules. So many of the hormones in our body are derivatives of lipids. So we'll think about a couple examples of those. Some important ones might be like estrogen and testosterone. They are lipid derivatives. So we take fats, we convert them into these testosterone and estrogen molecules, and they're important for signaling things in our bodies. All right, so let's take a look at the structure of a lipid. So with the macromolecules, we've been talking about the monomers that make up each macromolecule. Lipids are a little bit different in that they're not vast polymers of a repeating monomer. Rather, lipids are more discrete molecules. Lipids are made of up to 3 of these things called fatty acids. So if I had to assign a monomer, I'd say, well, there's fatty acid monomers. But we're going to see that we're never going to combine more than 3 fatty acids together at a time. So part of our lipid molecule will be these fatty acid monomers. And then another part, we're going to attach those fatty acid monomers to something called a glycerol molecule. So we'll take up to 3 fatty acids and we'll stick them on a glycerol, and that makes a lipid. Okay, so let's look at the structure of these fatty acids and the glycerol. So we'll start with the fatty acids. Here's a image to depict the structure of a fatty acid. This one shows it a couple different ways. We've got this line diagram or this ball diagram of the atomic structure. So a lipid is a long chain, this jagged line is a long chain of carbons and hydrogens. And it's nothing but carbons and hydrogens. Okay. So again, each one of these changes of direction in the line represents a carbon molecule, and there's hydrogens attached to it. So in the ball diagram, the black balls are the carbon, and the lighter color balls are the hydrogens. So we've got lots of carbons with hydrogens on them attached into this long chain. That's what a fatty acid is, a long chain of carbons and hydrogens. Sometimes those fatty acid chains bend. Like look at this one on the right. It makes a little bit of an angle here and goes a different direction. That's when there's a double bond in between 2 of the carbons in there. And so we classify fatty acids based on whether or not they are straight, they have no double bonds, or if they're bent, they do have a double bond. We name them saturated versus unsaturated fatty acids. So let's look at another diagram to really drive this point home. Here again, I'm showing you two fatty acids, one on top, one on the bottom. We can see that again, fatty acids are long chains of carbons and hydrogens right here. And sometimes they're bent. So the one on the bottom has this double bond, which causes it to bend. So again, saturated means there are no double bonds in here. Every carbon is bound to as many hydrogens as possible. It's saturated with hydrogens. Whereas unsaturated means there is a double bond in there. And you can see that these two carbons are only bound to one hydrogen each. So that's why we call it unsaturated. It's not saturated with hydrogens. So two kinds of fatty acids. They can be straight, saturated, or bent, unsaturated. All right. So again, when we make a lipid, we're going to take up to 3 of those fatty acids, and we're going to connect them together by attaching them to a glycerol molecule. So let's look at a glycerol molecule. A glycerol molecule is really simple. It's just a 3-carbon molecule that also has these hydroxyl groups attached to the carbons. These hydroxyl groups are really easy to attach stuff to. The hydroxyl groups are very chemically reactive. So what's going to happen with our glycerol is that to each one of these hydroxyl groups, we can add a fatty acid or something else. So we're going to take this glycerol, we're going to hang fatty acids off of it. Just to drive home the point, here's another diagram of a glycerol molecule, again, with balls representing the individual atoms. And again, we can see it's a 3-carbon atom, and there are these red oxygens. Again, it's the oxygens where we can attach stuff to the glycerol molecule. So let's look at some specific examples of kinds of lipids. All right, and the first lipid I'm showing you is what we call a triglyceride. Tri just means 3. Okay, so it's a 3-glyceride, means it's a lipid that has 3 fatty acids attached to it. And so this ball model is showing us the 3 fatty acids. They're each running from left to right, and they're attached to that glycerol molecule over here on the left. Whoops. So again, we take one glycerol, we attach things to it. In this case, we've attached 3 fatty acids. That makes what we call a triglyceride. All right. So let's look at the structure of another kind of lipid. This is one we'll talk a lot about in our course. It's called a phospholipid. So we've got this name phospholipid, right? And here's a diagram of the phospholipid. So right here in the middle, you can see this carbon, carbon, carbon. That's our glycerol molecule right there. Again, we can attach 3 things to the glycerol molecule. In this phospholipid, we've attached 2 fatty acids. Those are the 2 things hanging off here. Again, long chains of nothing but carbon and hydrogen. So 2 fatty acids, and then to the 3rd carbon of the glycerol, instead of adding a 3rd fatty acid like we did in the previous thing, the triglyceride, instead of doing that, we've added this phosphate group up here. So that's why we give it the name phospho, because it's got a phosphate group right here, and lipid because it's got the fatty acids down here. So phospholipids have 2 fatty acids and 1 phosphate group attached to that glycerol molecule. All right. The last kind of lipid I want to show you is something we call a sterol. So here I'm showing you 2 different images of a sterol. And sterols are weird because instead of having that long straight chain of carbons and hydrogens, we've sort of twisted the chain up and we've created these rings in here. So sterol molecules have these ring structures, but they're still derivatives of those long fatty acid chains. Again, we've just modified them. We sort of hooked them back onto themselves to make these ring structures. So those are the 3 kinds of lipid structures we're going to talk about. And just to wrap up, let's think about the specific functions that each of those lipid molecules performs. So those triglycerides, again, that's the one where we have 3 fatty acids attached to the 1 glycerol. Triglycerides are what we use to store energy. So if we could examine the body fat on our bodies, it would be made up of triglycerides. All right, the phospholipids, Again, that one has the glycerol. It has 2 fatty acids attached to it, and then 1 phosphate group. Those are critically important for making up the cell membrane. So as we talk about cell structure, and specifically the cell membrane moving forward, we're going to talk a lot about phospholipids and why they function the way they do. They're really important for cells. And then the last structure I showed you, those sterols, where the fatty acid chain has been linked to itself to create these rings. Those sterols are what we convert into signaling hormones, things like estrogen and testosterone, cholesterol. So 3 different kinds of lipids perform 3 different kinds of functions. All are critically important to cells, and that's why we care about lipids in biology. All right, so we'll end there. As always, let me know if you have any questions, and I will talk to you again soon.